Hello everybody, and welcome to this talk titled Pancreas Volumetry in UK Biobank, Comparisons of Models and Inference at Scale. My name is James, and I work in the data science team at Perspectum. Today I'm going to talk to you about a piece of work looking at how we can more accurately measure pancreas volume from UK Biobank data and present some interesting findings from this data. Hopefully this presentation will demonstrate how we can leverage these massive datasets alongside deep learning based segmentation to produce some clinically meaningful results. First of all, why do we care about pancreas volume? Pancreas volume has been shown to change with age and in diseases such as pancreatitis, type 1 diabetes and type 2 diabetes. If we can detect these changes in some longitudinal health study, it may be a useful early indicator of disease. We can also, assuming that we've calculated pancreas volume from a mask, a segmentation mask of the pancreas, we can also perform shape-based analysis to investigate new biomarkers, or we can automatically extract quantitative metrics from those segmentations, such as MR-PDFF. I'm sure a lot of you are familiar with UK Biobank, but as a quick reminder, UK Biobank is a major health resource that contains in-depth genetic and health information for 500,000 UK participants. The data collected from an individual will be at multiple time points, with the idea that if that individual, and it's also linked to their health records, so if that individual goes on to develop some type of rare disease in the future, we can retrospectively look back at all of that individual's data to try and disentangle why that person went on to develop the disease in the first place. The part of UK Biobank contains a imaging sub-study which aims to conduct detailed MRI scans of over around 100,000 participants, making it the largest imaging study in the world. Part of this MRI data in UK Biobank includes a high-resolution 3D MRI scan that specifically targets the pancreas region. The high-resolution nature of this scan can allow models to learn useful representations of the pancreas, and we can also better quantify biomarkers such as surface lobularity, which is looking at how smooth the surface of the pancreas is. However, as we started to look at these scans, we noticed that a lot of the acquisitions only had partial coverage of the pancreas. So we thought, what can we use instead? And also within UK Biobank, there are these whole body neck to knee Dixon vibes. So we looked into if we could use these whole body scans to more accurately measure the volume of the pancreas. So you can see on the left-hand side here, there's an example of the pancreas-specific scan. On the right-hand side, we can see an example of the full neck-to-knee scan. And we can see that the pancreas-specific scan on the left misses a fairly large chunk of the pancreas head. And in this case, it led to an underestimation of pancreas volume of around 14%, which would be clinically significant. So what is our proposed pipeline? Here's a rough outline, but it takes this stitched full body neck to knee scan. We then take that stitched image and use rigid image registration to move the input image into a fixed reference space. And why do we want to do this? We do this to roughly align where the organs are within that image, so we can then heuristically crop out the abdominal region. Why do we want to crop out the abdominal region? It's essentially to save GPU memory, to avoid downsampling the image to fit into memory. And it's also more efficient. As you can imagine, regions around the thigh are not going to contribute meaningful representations, good representations of if we're just interested in the pancreas. So we then crop out the region, perform automated pancreas segmentation on that cropped 3D region, and then apply the inverse transformation to get back to the original space and extract a pancreas volume from that. In terms of the segmentation model that we chose to use, we wanted to run this pipeline to segment 42,000 pancreas, hence the need for automation in this case, and it also increases reliability and decreases subjectivity of the segmentations. So we were inspired by two different papers, one from A.I.L et al. And one from Octet et al. The one from Octet et al. Introduced the idea of attention gates within UNet. The idea being that these gates are a mechanism such that the network can learn to focus on features that are relevant to the task. And the residual unit and recurrent residual unit were introduced by A.I.L et al. Have the idea that the residual connections can help improve optimization during training, and the recurrence units, the recurrent layers, the idea behind those is that features are accumulated in a layer by forward propagating through that layer multiple times. We also combined the residual unit and the residual recurrent unit with attention gates to see if we can utilize each of the potential benefits from each network. And you'll be able to find more details about the networks in the full paper. In terms of model training, we had 200 expertly labeled images, of which we split into 70% training, 20% testing, and 10% validation, using the same training and testing and validation split for each model each time we ran tests on the model. We also applied slight data augmentation and had a loss function of combined dice and cross entropy, which we found to perform slightly better than just using either a dice loss or just using a cross entropy loss. We also used atom optimization and mixed precision training, which helped to reduce the memory requirements for the model, again, so we could fit a larger image into the GPU memory, and it sped up training time and inference. So here you can see the model performance results. Each model performed quite similar, with no statistical difference between the performance of each model. We chose to go with the attention unit to run inference to extract the pancreas volume for all of the subjects, just because it seemed more susceptible to less susceptible to outliers compared to the other types of model. Here you can see a couple of figures. So on the left hand side, we can see the distribution of pancreas volume for both males and females. The values in terms of the median volume are in line with previously reported values in the literature from smaller studies. But to the best of our knowledge, this is the largest pancreas volume study to date. On the right hand side, we can see an interesting figure showing diurnal variation of pancreas volume. And what do I mean by this? So diurnal variation refers to how the pancreas changes in size throughout the course of the day. So you can either measure a single individual at multiple time points throughout the day and see if that individual's pancreas stays the same size throughout the day, or you can scan thousands of individuals at just a single time point throughout the day and look at the average volume for each distribution. So we've rounded the time at which a subject was scanned to the nearest hour and then plotted, you can see the median pancreas volume throughout the course of the day. And interestingly, the pancreas decreases in size throughout the day. We're not entirely sure what causes this, but it's supposedly linked to your circadian rhythm, so your sleep-wake feed fasted cycle. And this could be important if you're a clinician about to make a decision, a clinical decision based on pancreas volume. So we suggest that this change in volume throughout the day should be considered when making these decisions. Thank you for listening.